Hello everyone, I am Dr. Manisha, Postdoctoral Fellow, ISR Mohali. I thank the organizers for giving me an opportunity to represent my research at this international symposium on recent advances in self-assembled materials and supramolecular chemistry. The title of my poster is Label-Free Detection of Ochratoxin A using aptamer as recognition probe at liquid crystal aqueous interface. Why we choose the ochratoxin A? Ochratoxin A is a natural occurring mycotoxin produced by several fungal species and most widespread food contaminant. It is mostly present in cereals, food juices, spices, etc. The Internal Agency for Research on Cancer IARC identified OTA as human carcinogenic agent. The presence of OTA even at a very low concentrations in agricultural products and foodstuffs may cause serious toxic effects. Therefore, the detection of OTA in foodstuffs and agricultural products is of prime importance to food safety, quality control and human health. However, there are many conventional analytical methods to detect OTA, but these techniques impose some drawbacks which restrict their practical applications such as time-consuming, expensive or complex instrumentation, complicated pre-treatment of the sample and it requires trained operators. Therefore, we get motivated to design a simple label-free platform for the detection of the ochratoxin A. For this, we use the liquid crystal aqueous interface as a label-free platform to design the sensor for the detection of OTA. Now the question arises why we choose the liquid crystal as a platform to design the sensor and what is the liquid crystal. Liquid crystal is a intermediate state between crystal and liquid having a orientational order. Due to this orientational order, LC molecules tend to align themselves along a particular direction either perpendicular or parallel, which gives the different optical appearance under polarized optical microscope. Either when they align themselves as a perpendicular direction, they give the dark appearance which is known as the homeotropic orientation, and when they align themselves in the parallel direction to the substrate, they give the bright appearance which is known as the planar orientation of the LC. For the designing of the LC sensors, we commonly use the LC which is 5CV. It is a 4-cyano-4-pentyl biphenyl system and it is a room temperature LC system. Next, LC interfaces are very sensitive to their external environment, which we can see here in this figure. When LC is in contact with air, it gives the homeotropic orientation, and when it comes in contact with the water molecules or aqueous system, it gives the planar orientation, and when it comes in contact with some other surfactant molecules or lipid molecules or other molecules, it changes its orientation back to the homeotropic orientation. So we can say these. Therefore, these LC aqueous interfaces are highly dimorphable and used to study various biological events such as protein adsorption, antigen antibody binding, enzymatic reaction and so on. These LC based sensors are very simple, easy to handle. They offer label-free detection by the naked eye and they do not require any complex instrumentation. Even a smartphone can be used to perform LC based sensing in a remote area. Nowadays, to increase the sensitivity and selectivity of LC sensor, the use of aptamer as a molecular recognition probe has attracted the considerable attention. Why aptamer? Aptamer offers exceptional advantages such as strong affinity toward a particular target and numerous advantages over immunosensors such as robust stability in aqueous solution, simple design and cost-effective synthesis. Therefore, we use the aptamer sequence which is selected by Cruz Aguado et al., which is OBA wild type of aptamer, and it binds with OTA with the dissociation constant in a micromolar range. OTA could induce or stabilize G quadruplex formation of a aptamer sequence and thus it could enable detection of OTA with high sensitivity and selectivity. Therefore, we integrated the aptamer with LC aqueous interface to design LC apta sensor. For this, first we made the LC film by introducing 5CV onto the gold grid spotted on the DMOF coated glass slide. DMOF is known to align the LC molecules perpendicular to the substrate, which gives the homeotropic orientation of the LC and dark appearance under cross polarizer. On the introduction of CTAM molecules, the CTAM molecules will get self-assembled at the interface through the hydrophobic interaction between the CTAM chains and the LC molecules. Further, when we will introduce the aptamer to this system, the aptamer will adsorb electrostatically to the CTAB-laden LC interface and it will disturb the self-assembly of the surfactant. Further, on the introduction of the OTA, OTA will bind specifically with this aptamer and it will induce a change in its structure in terms of conformation or helicity of the aptamer, which ultimately release the CTAB molecule to self-assemble again at the interface and it will give the again homeotropic orientation of the LC molecules. Based on our proposed design, our first set of experiment was designed to optimize the concentration of CTAB to obtain the homeotropic orientation of the LC molecules. For this, we have changed the concentration of the CTAB from 0 to 9 micromolar with respect to time, and we observe that we got the complete homeotropic orientation of the LC at 7 micromolar concentration. It means at this particular concentration, CTAB molecules get self-assembled at the LC aqueous interface. Therefore, for the further experiments, we use this 7 micromolar concentration of the CTAB. Now, our next experiment was to optimize the aptamer concentration, where we will get the planar orientation of the LC. So, here also we vary the concentration of the aptamer from 300 to 50 nanomolar, and we observe that at 100 nanomolar the complete planar orientation of the LC was observed. Below this, the planar orientation was not stable. Therefore, this 100 nanomolar concentration is the optimized concentration for the further experiments, and at this particular concentration, CTAB molecules get deassembled from the LC interface and it binds with the aptamer through electrostatic interaction, as discussed in the previous slide, that CTAB decorated LC aqueous interface gives the homeotropic orientation due to the hydrophobic interactions between the tails of the CTAB molecules and the LC molecules, which on the introduction of the aptamer forms the insoluble complex with the CTAB molecule in the aqueous medium. The electrostatic interaction between the surfactant and the aptamer and the hydrophobic interaction between the nucleobases and the LC disturbs the self-assembly of the surfactant molecules at the interface. This results in the decrease in the surface coverage of the surfactant at LC aqueous interface and trigger the reorientation of LC from homeotropic to planar. In the presence of the OTA, the aptamer binds with the OTA, which changes the conformation of the aptamer by forming the G-quadruplex structure, which increases the surface coverage of the surfactant molecules at the interface, and the homeotropic orientation has been observed. In order to examine the conformational changes in the secondary structure of DNA in presence of OTA, we recorded the CD spectra of CTAB and pre-incubated mixture of aptamer and CTAB in presence and absence of the OTA. In presence of OTA, a change in ellipticity has been observed that reveals the stabilization of G-quadruplex conformations of the aptamer in presence of OTA. These results were further supported by the THT. THT is a non-emissive water-soluble dye which is known to bind selectively with the G-quadruplexes and result in the significant enhancement of the fluorescence intensity. As we can see in the graph that THT is itself a non-fluorescent, but in the presence of aptamer, it gives the highest fluorescence. But on the introduction of the OTA to the above THT aptamer solution, the fluorescence intensity of the THT decreased significantly, which confirmed the release of THT upon the competitive binding of the OTA into the G quadruplex, as we can see in the schematic representation also, that THT gets displaced from this G quadruplex and OTA takes place. Next, we were interested to investigate the sensitivity and the response time of LC-APTA sensor toward the detection of the OTA. For this, we vary the concentration of the OTA from 250 nanomolar to 0.01 nanomolar, and we observed that at higher concentration of OTA, the homeotropic orientation of the LC was observed within 1 minute. However, on decreasing the concentration of the OTA, the response time of the LC-APTA sensor gets increased. For example, for 1 nanomolar and 0.1 nanomolar, the response time of the LC-APTA sensor was found 30 minutes. Therefore, based on these experiments, the detection limit of the LC-APTA sensor was found to be 0.1 nanomolar. For further quantitative analysis, we measured the average grayscale intensity of the optical images captured at different time for the different concentration of the OTA, and we observed that at higher concentration, the GI value decreased immediately within 1 to 5 minutes. However, further decreasing the concentration of the OTA, the GI value decreased slowly. For example, on further decreasing the concentration, for example 0.01 nanomolar of the OTA, there was no significant change in the GI value has been observed. Next, then we plotted the graph between average grayscale intensity and the log value of the OTA in the picomolar range. We observed the linear correlation between GI and logarithm of the OTA concentration between this range, and therefore we can use this range for the detection of the OTA, and the detection limit was found to be 0.1 nano. Next, we were interested to see the effect of the pH towards the detection of the OTA using LC-APTA sensor. For that, first we see the effect of pH on C-type self-assembly. We observe that in presence of 7 micromolar C-type, LC remain homeotropic at pH 5 as well as pH 9. However, in presence of aptamer, the homeotropic orientation of LC triggers to planar orientation at pH 5. However, in the pH 9, we observe the mixed domain of planar and homeotropic orientation. Therefore, we perform the experiment for the detection of OTA at pH 5, and we observe that the planar orientation of the LC changed to the homeotropic, but it takes the time. It means the detection of OTA is slower at pH 5 in comparison to the 7.4. Next, we perform the experiment to see the effect of ionic strength of the Happus buffer in range of 1 to 100 millimolar. We observed at 1 to 100 millimolar range of ionic strength of Happus buffer, LC remain homeotropic in presence of 7 micromolar C-tab. However, in presence of C-tab aptamer complex, LC shows the planar orientation at 1 to 30 millimolar buffer. However, at higher buffer strength, the coexisting of bright and dark LC domains were observed. Higher ionic strength of the buffer screened the electrostatic interactions among cationic surfactant molecule, which lead to a tightly packed surfactant monolayer at the aqueous LC interface, which can't be disturbed by the presence of the aptamer. Therefore, we use the 1 to 30 millimolar Happus buffer range for the detection of the ochratoxin using LC-APTA sensor, and there is we detected the ochratoxin at 10 millimolar buffer strength of the concentration 250 nanomolar and 100 nanomolar. Similarly, at 30 millimolar of the buffer strength, we use the ochratoxin concentration 250 nanomolar and 100 nanomolar, and we observed the homeotropic orientation after 30 minutes. After this experiment, it was observed with increasing the ionic strength of the buffer, the response time of the LC-APTA sensor also increased. High sensitivity of LC-APTA sensor motivated us to investigate the practical application of designed LC-APTA sensor for the detection of OTA in real samples. For this, we spiked the tap water and apple juice with 250 nanomolar OTA. We observed that LC remained homeotropic when 7 micromolar C-tab was incubated with tap water and apple juice. In presence of pre-incubated C-tab and aptamer and tap water or apple juice without OTA, the LC exhibits the planar orientation, which changed to the homeotropic when the tap water or apple juice spiked with the OTA. This result suggests that the designed LC-APTA sensor can be used for the detection of the OTA in the real samples. Now, conclusion of the talk. We developed an LC sensor for detecting ochratoxin A using OTA-specific aptamer as a molecular recognition probe, and we found the detection limit was very low as of 0.1 nanomolar. We proposed the mechanism on the basis of the CD and THD assay. We also studied the effect of pH and ionic strength of the buffer toward the detection of the OTA using this LC-APTA sensor. We also demonstrated the practical applicability of the LC-APTA sensor for the detection of OTA in the real samples. We have published this work in the Frontiers in the Soft Matter. It is a just accepted acknowledgement. I acknowledge the IISER Mohali for providing research infrastructure and postdoctoral fellowship. Thank you.